Okay, so let's take a cell like that with its solutes inside. Remember, the solutes can't escape because they can't get through the cell membrane. And as a thought experiment, we're going to put it in a beaker of pure water. Now, by the principle of osmosis, where's the water going to go? Into or out of the cell? Into the cell, right, because the water is attracted to the solutes, water follows solutes is the rule, and so water is going to rush inside there. Now, any kind of solution like that where you have a lower solute concentration than a cell is called a hypotonic solution. Hypo means less than or beneath, and you can think of tonic as meaning the solute concentration of the cell. So literally it's a solution that's lower solute concentration than the cell cytoplasm. So anytime you place a cell in a hypotonic solution, such as pure water, water will always rush into the cell by osmosis. And typically what that's going to mean is that the cell is going to expand. If you think of the membrane as being somewhat soft and flexible like the rubber skin of a water balloon, it's just like adding more water to the water balloon. And so this one will get larger and larger and larger. And I don't have it here on the cartoon, but in some cases the cell can literally explode like an totally overinflated water balloon. The membrane just tears and kablooey, that's the end of the cell. Now that doesn't always happen. It really depends on exactly how hypotonic the solution is and how long you let the cells sit inside there. The cells will always gain water, but whether or not they explode depends on the solution. Now we're going to flip the experiment around and we'll put the cell in a liquid that's hypertonic. Hyper meaning greater than, and tonic again meaning the solute concentration inside the cell. Now where's the water going to go? Out of the cell. It's still osmosis. It's still water following the higher concentration of solutes, but now that happens to be outside the cell. So in hypertonic solutions, cells do this. They shrivel and shrink. And I didn't make the cartoon as well as I should have. What they actually end up is looking more like raisins. As the cells lose water, they get convoluted and shrunken and shriveled. All right, so hypertonic solutions will cause them to lose water and the cells get smaller. Now let's do one more thought experiment here. Here the cell is going to be in a solution that has the exact same solute concentration as the cytoplasm of the cell. Now where does the water move? Nowhere, because osmosis only happens if one side of the cell membrane has a higher solute concentration than another. And that's not true in this situation. It's equal concentrations inside and outside. And so you're not going to get any osmosis in that sort of situation. And that sort of solution is called an isotonic solution. Iso meaning equal to, so equal to the cell's solute concentration. Now, if you read the textbook on isotonic solutions, they will tell you that in actuality, a few water molecules still do go in and out of the cell, but the point is it's equal numbers going in and out, so there's no net osmosis going on in the cells, therefore not going to change its shape. And if you think about it, that's a good thing, because cells don't want to be shrunken and shriveled the way they would end up in a hypertonic solution. That's just not healthy for them. And cells also don't want to be filled up with huge amounts of water, especially to the point where they explode. That obviously wouldn't be healthy for them. So cells are happiest and healthiest in isotonic solutions. And therefore, it's not too surprising to learn that your body fluids are isotonic solutions. Your blood and the tissue fluids that surround the cell, all of those are more or less isotonic solutions. Now, when a person goes to the hospital, sometimes they have to put fluids inside the person, like IV solutions and things like that. And when they do put solutions inside a person's body, they usually don't want those solutions to be hypertonic, because then you're putting the person's cells in an environment that's going to shrink their cells, and they don't want to put hypotonic solutions where they might explode the person's cells. And so almost always hospital solutions are isotonic solutions. And again, there's a few exceptions to that rule if they're, for some reason, want to add water to your cells or take water out of your cells. But by and large, they are isotonic solutions. You buy it so far? There we go. All right, well, let's see. This red thing at the top of the diagram represents one of your cells, a cell from a member of the animal kingdom. From the looks of it, it's supposed to be a blood cell, but it could really be any cell in your body. And so this shows that in an isotonic solution, there's no net osmosis. The number of waters that drift in are equal to the number of waters that drift out, so the cell keeps its normal shape. And this is the normal shape for a blood cell. But if you put that blood cell in a hypertonic solution, it loses water and gets all shriveled and shrunken. But if you put it in a hypotonic solution, it gains water, possibly to the point where it explodes. And the official term for a cell tearing and exploding is called lysis. The bottom row shows a plant cell, and because of the plant cell's rigid cellulose cell wall, you get a slightly different reaction in the cell. All right, so remember just like our cells, plant cells have a plasma membrane and they have membranous organelles and they have cytoplasm, but the difference is plants have this rigid cellulose cell wall that acts like a rigid cage surrounding the cell. If you put a plant cell in a hypotonic solution, water will rush in just like it normally does, but the cell can never explode because the cellulose cell wall keeps it from exploding. And plants actually like that sort of thing. Think about non-wooden plants like grasses and weeds and things. Everything else being equal, those plants want to be upright so they can catch more sunlight. They don't want to be flaccid and laying flat on the ground. And so they like to be in a slightly hypotonic environment to make themselves rigid and hold themselves upright. If you put plant cells in a hypertonic solution, water exits the cells just like you'd expect it to, but the cell wall doesn't change shape because it's a more or less rigid structure. And so you get sort of an interesting effect where the plant cell parts that other than the cell wall shrivel up and get all shriveled and shrunken, but the cell wall stays its exact original shape. And I might have some photographs of that. Yeah, you can see it here. These are plant cells. Here you can see the cellulose cell wall around like that. The little green things inside are those chloroplast organelles, which are where the photosynthesis takes place. Anyway, so here are the plant cells either under isotonic or hypotonic solution, where the water's rushing in or pressurizing them. But you put them in a hypertonic solution, the cell membrane peels off the edge of the cell wall, and the cell membrane and all the organelles shrivel up in the inside from water loss. Not this week, but I think next week in lab you'll be doing some labs on osmosis and diffusion. And you will do this to plant cells. It sounds kind of cruel almost, but anyway, it's kind of a neat demonstration of the osmosis principles. All right, a little question here. So if I took this cell right here and put it in this solution, which has a higher solute concentration, the cell is in hypertonic, hypotonic, or isotonic conditions? Okay, hypertonic, right, good. And, oh, I clicked it too soon. I was going to say, where's the water go? The water exits the cell. And so the cell is going to, well, this is obvious, the cell is going to shrink or grow. Shrink, right, just like a leaky water balloon. It's going to get smaller and smaller and get all shriveled. Alrighty, well, you guys now are experts in osmosis. Let us continue with the energy and metabolism. So one of the big pictures that we're following in this chapter is how cells store up energy. And the first way we learned about was forming a concentration gradient across the cell membrane like you see there. The other way cells store potential energy is something called chemical potential energy, which is also think of as making high calorie molecules. Let's see. Here I define the chemical potential energy this way. It's the energy stored in a molecule's covalent bonds. Let's use the glucose molecule as an example for a moment. Molecules are made out of atoms that are joined together. Like the glucose there is made out of 6 carbons and 12 hydrogens and 6 oxygens all joined together. And the forces that hold the atoms together, that glue them together, are bonds, in this case covalent bonds. As it turns out, there's potential energy stored in each covalent bond. Each and every covalent bond you can think of as almost like a little battery that holds some stored up energy in it. And so everything else being equal, the more covalent bonds in a molecule, the more energy is stored in there. And the term calories is a unit of energy, and it's typically applied to the amount of energy in the foods we eat, but what it really comes from is the energy that's stored in the covalent bonds of the foods that we eat. There are differences in the chemical potential energy, the stored energy, in different types of covalent bonds. And for this lecture, we're mostly going to focus on the bonds that connect to carbon atoms, just because we focus on organic molecules when we talk about the molecules in living things. And here's the hierarchy. It's there in your notes, but it's also up there in the digital screen. The carbon-hydrogen covalent bond has the highest energy stored in it. Carbon-carbon covalent bond is medium energy.